Okay, this video is going to be on mutations. And mutations are really just it's a random change in DNA. So how and when can this kind of thing take place? Well, one of the things that can happen is we can have a copying or an editing error that happens during DNA replication. We could also have like some environmental factors. And these environmental factors could be things like, well, X-rays, UV light, chemical, various chemicals. It always seems like there's something new that's being considered to be a carcinogen, for instance. So all of these things will lead to this random change in DNA. And again, remember the ultimate score is that we're looking for variation. You know, we want to have some variation in our species. Okay? Again, I want you to think that this is a pure random event. I can't emphasize that enough. It is not directional. It is not trying to find the best fit. It is just random. And sometimes it does find the best fit by dumb luck, by chance. But it is not directional. Okay, and of course, all of these things need to be inherited. So how can they be inherited? Well, if we're talking about like some sort of body cell, every cell that comes from that cell, so if there's a mutation, every cell that comes from that cell all those cells will also be mutated. So if you have a mutation, all cells that come from that cell that is mutated will also be mutated as well. However, this does not affect the next generation. Important to know that. Your skin cells, those genes that are mutated in skin cells, cannot be passed on to your children. Another thing that can be passed on are the sex cells. And that brings me to the second one. Gametes. These mutations are passed on to the next generation. Okay, why? Well, these are the sex cells. Okay, these are the sex cells. These are what are going to make the next generation. Okay, so let me get just a little bit of some room here. So if you don't mind, I'm just going to wipe. Much better. Okay, so what happens when we have a mutation? There's a series of steps that kind of take place. And I really want to walk you through those because we're going to keep bringing this up again and again and again. So first, we mentioned there's a random change in the DNA. And what are we talking about? The DNA sequence. So we're talking about the A, the T, the C, and the G, all those letters in a row. So that change in the DNA sequence will lead to it'll lead to a change in the mRNA sequence. And the mRNAs then go out to the ribosomes, and this will cause a change in the amino acid sequence, the order of the amino acids that will make up the protein. Okay, so let's stop for 1 second here. I want you to notice here, if we have a change in the DNA sequence, that's definitely going to change the mRNA sequence. But will it always change the amino acid sequence? Will it always change that amino acid sequence? Because it's quite possible that a difference, if you remember that there are 20 amino acids, but there are 64 different codons. So that means there's more than one 3-letter mRNA sequence that will make one amino acid. So therefore, it's possible that this is what we call a silent mutations. Possible we have a silent mutation. Just simply means that it didn't change the amino acid sequence because 2 different mRNAs could make the same amino acid. Just throwing that out there. Keep that in mind. So if we change the amino acid sequence, we'll also change the protein structure. Because you remember, we changed the protein structure, it changes how the protein will, if you change the amino acid sequence, you change one of those amino acids that like to be on the outside, but now it wants to be on the inside, and suddenly that protein folds up much differently. Well, if we change the protein structure, most definitely going to change the protein function. If it doesn't have the same shape, it may have a different structure. Let's go back again. Is it possible that even though you have a different amino acid sequence, that it will still fold up the same way? So again, yeah, another silent mutation is possible, meaning that we could have a mutation that really has no change in the protein structure or the amino acid sequence. Silent mutations. So if we change this protein structure, we change the protein function, what it does, it may change the cell, the tissue, the organ, their structure, and their function. Structure determines function. So therefore, if we've changed this protein function, it may change what the cell, what it looks like, its structure, what that cell does. Great examples coming up, and we're going to talk about sickle cell anemia. So, pay attention to that one. It's a real good example of all of this stuff. Got all this? 'Cause I need some room. So what are some of the types of mutations? Okay, so there's 4 basic types that we're going to talk about. The first one is called a substitution. Okay, substitution mutation is just like it sounds. You're going to take one DNA base and you're going to substitute it for another. So let's imagine we have a sequence in DNA C, T, G, G, A, G. And that's our DNA normal sequence. But in a substitution reaction, perhaps we do something like this. C, that's a C. C, T, G, G, T, G. So notice we have a substitution right there. So what kind of things could this have happened? We could have a change in one amino acid. Okay, because that 3-letter sequence right there, that code, which eventually makes a codon. Or we could have like no change. And of course, we could also have an incomplete protein, which is, kind of the surprising thing. Because what happens if this changes in the middle of the DNA sequence, in the middle of the genes, and all of a sudden, well, this actually, one little change causes for a stop codon, and that stop codon then causes the protein to be incomplete and not completely made. Okay, so another reaction or another type of mutation, 2, is called an insertion or an addition. As just as it sounds, we're going to add more DNA. So again, here's our sequence, this CTGGAG sequence. That's our normal. And well, maybe we have an additional piece in here. So maybe we have TTGG. So notice that these letters are inserted or added in to our sequence. Okay, and again, it could just add up an amino acid, or it could lead to some other changes. I mean, it doesn't always have to be in groups of 3. If it's in groups of 2, it could cause another stop codon later on. Okay, our third one. You know, if we can have an addition and an insertion, obviously we could have a deletion as well. Okay, so a deletion, just as it sounds, we're going to take our normal sequence, CTGGAG, and we're going to remove some of the bases. So we're going to have maybe a CTAG. We remove these 2 middle G's right in there, these right here. Okay, all right, and again, we could really cause a major change. Okay, and our last one, and probably a most harmful one, is called a frame shift mutation. Now, what do we mean by this frame and shifting this frame? So I want to talk, you know, we think of the frame as being 3 letters. Well, why 3 letters? Well, that eventually makes the codon. And the codon tells us which amino acid. Okay, this is again on mRNA, and the codon determines the amino acid. Well, they come in 3 letters. So I want you to imagine, let's say I have this little phrase here. The fat cat sat. Well, let's say I delete the first letter. Well, now it's really hef, at. That's like gibberish. It means nothing. It's exactly what happens in a frame shift mutation. So if we were to change our sequence, let's take our normal here, CTGGAG, and we add an extra base in there, or let's say we add an extra one. If we go CTGAGAG, well now, here's one amino acid, here's the second amino acid. Well, here's our new problem. We got this amino acid, we got this. Notice the second amino acid well, I shouldn't say amino acid. Our second frame is completely different from the first one. It's going to make a completely new mRNA, a completely new amino acid, completely different. So this usually leads to some major issues. Okay? All right, so overall, mutations. I want you to realize, again, that they are a random thing. Okay? I'm going to slide over here. Okay? Mutations are random. Okay? They can lead to no change. We mentioned that silent mutation. They could lead to only a slight change. Maybe one amino acid difference. They could lead to a major change, like coming up with an incomplete protein, or perhaps even like this one, a frame shift mutation. And even more so, we could have a lethal change, which means the death of that organism. Okay, so that covers mutations. Hope you learned something.